talks and a whole gamut of different things, just trying to get people interested in science, and scientists communicating with the general public about what they do. I think that's really important these days. And so we have a number of talks, and Polly was really, Polly Matzinger was generous to call us up and say, hey, she'd like to give a talk here. And I'd like to welcome her to BUGSS.
Thank you. Thank you. I can decide after if you still want to do that. So usually when I talk to people, I tell them I have one rule, and that is don't fall asleep. And the reason I make it a rule is that I figure it's your fault, not mine. And here's why. You fall asleep if I use a word you didn't understand. Or I use a word you did understand, but you didn't understand how I used it, and so you get confused. Your brain doesn't like confusion, and so you get kind of woozy, and then you get more and more confused because that first confusion kind of underlays a whole bunch more. And then you go to sleep. So it's your responsibility if you start feeling a bit woozy to right then and there ask me the question you need to ask to get unconfused, so you don't fall asleep. Now, I enforce this, I throw things. And I have a bad aim. So it's a good idea in self-defense to wake them up and get them to ask me that question, right? So don't wait till the end. Stop me right then there if something doesn't fit. If you know something doesn't fit with what I'm saying and you're kind of thinking about that, bring it up. Okay? Okay. So my next question, then, my first question is, how many of you are not immunologists? Not. You're an immunologist back there in the peak?
Not. Innate immunologist.
Okay, an innate immunologist is a big deal. How many of you have not had an immunology course either? Okay. So those of you who are, or who have had an immunology course, were probably taught what I was taught as a graduate student, which is that the immune system functions by discriminating between self and non-self. That it learns what self is. Each individual's immune system learns what self is early in life. So anything that's around in utero or just after birth, a couple days after birth, is labeled as self. You don't fight that. Anything that comes later, viruses, kidney transplants, whatever, is labeled as non-self, and you do fight that. Okay? So, were you taught that? Yeah? Okay, so was I. And I was in graduate school in the 70s, before most of you were born, and I was taught that, and I had terrible trouble with the idea, because there were all kinds of things that didn't fit. So I asked my professors, for example, what happens when you go through puberty? You know, we're not the same sitting in this room as we were in utero or a couple days after birth. And they patted me on the head and said, you're there. Nothing new appears at puberty. It's just changes of concentrations of hormones and things. And so I pointed out a condition to them that they hadn't thought about, mostly because they had white and that is this tissue. Right? It starts making proteins later in life. The cells that are making those proteins secrete them. And although most of them go into the outside world, the newly lactating breast makes at least 2 proteins that end up in the bloodstream. One is alpha-lactalbumin, and the other one is beta-lactoglobulin. And they end up in the bloodstream. And the cells that make these proteins display peptides from those proteins on the surface in special sugar molecules called MHC, which, by the way, are the molecules that you have to match for when you do a transplant. So almost all the cells in the body put up a window display on their surface of what they're making inside. And it's a way for the immune system to see a virus, for example. The virus gets in, the viruses would like to hide, but there's this machinery that says, you don't get to do that. We're going to put little pieces of you on the surface so the immune system can see it. And cells do that most of the time. I sometimes say all, it's not true, red cells don't do this. But they don't have a nucleus, they can't really get infected by a virus. So they don't need to do that. Okay, makes these proteins, and by the self-non-self model, since these things appear later in life, it ought to be rejected. But it isn't. And so when I brought this up to my professors, they said, oh, well, that's because when this individual was learning self, she was drinking mother's milk and becoming orally tolerant. And so I pointed out to them that my generation actually has done an experiment. When I was born, in the late 40s, it wasn't socially cool to drink mother's milk. Right? You drank Nestle's or Carnation and Pet milk. These companies are still trying to do this to third world women. The point is that when my generation grew up and started lactating, our breast didn't fall off. No. Somehow, the immune system knows how to deal with a changing self. Males have the same problem. Right? You start making sperm as a teenager. You never made them before. And yet, your testes aren't rejected. Immunologists, for years and years, not worried about the breast, but very worried about testes, suggested that the testes are immune privileged sites where you don't have any immune response.
Right on.
And by the way, I believed that for 30 years. It's still written in textbooks that you have these immune privileged sites where the immune system just doesn't go. And about 2 years ago, I caught myself and said, wait, hang on a second. Imagine a tissue which is wet, warm, and full of nutrients. If it weren't immunologically protectable, don't you think the pathogens and the parasites would have figured it out pretty quick, because that's where they're going to go? Well, the eye is a privileged site. The testes are privileged sites. They don't get more infected than other places. And I now think that's a completely wrong idea. And if you want to invite me back later, I can tell you about that, but we're not going to talk about it today. All right? So one of the problems with the self-non-self model is the unilactating breast and the newly functioning testes. Another problem with the self-non-self model, which a colleague of mine, Charlie Janeway, called the immunologist's dirty little secret, is the additives that you have to add to vaccines to make them work. So if you make a vaccine using a virus that you've attenuated or killed, often it works, right? You just need the virus. But if you want to make a modern vaccine, a designer protein or peptide vaccine, most of the time they don't work unless you add something to it called adjuvant. Right? So for example, if you take ovalbumin, which is egg white from chickens, and you inject it into a mouse, it's very definitely foreign to a mouse. Right? It doesn't matter how you inject it. You can inject it intramuscularly, subcutaneously, intracranially, intravenously. You don't get an immune response, even though it's foreign. Unless you bought it from Sigma, and it has LPS in it. LPS is a bacterial molecule that the immune system responds to. To get a response to ova, you need to add adjuvant, and our favorite adjuvant is a mixture of mineral oil and mycobacteria, the bacteria that give TB, except we use bovine mycobacteria. And it's called complete Freund's adjuvant after the guy who made it. If you mix the ovalbumin in Freund's, now you get a nice response. That's non-self. The same is true of self. If you take myelin basic protein, which is a brain protein, from a mouse, and you inject it into a mouse, not a mouse. If you mix it with complete Freund's adjuvant, now you get a nice response. If you add something really nasty, like pertussis toxin, pertussis gives you whooping cough, or used to before we had vaccines. Okay. You add the toxin from that, now you get a disease called experimental autoimmune encephalomyelitis, which people use as a mouse model of multiple sclerosis. You turn on the immune system to attack myelin-basic protein, and it takes the myelin off of your nerves, a mouse's nerves, and they get paralyzed. So it doesn't seem to matter to the mouse whether you inject non-self or self. What matters is you mix it with adjuvant. So this has been known for a long, long time. But you know, one of the things that happens to us scientists and to you scientists is if something doesn't fit with your model, you tend to ignore it. So it was, these things got ignored. There are lots and lots more problems that I had when I was a graduate student, but I don't have time to be here for 6 hours. And you know, those are just a couple examples. So what happened was, when I was a graduate student, there wasn't a lot of support for asking these questions. And there was like zero support for trying to answer them. So I did the other thing we cowardly scientists often do. I stopped asking. And I went to do a 4-year postdoc in Cambridge, England, and then a 6-year postdoc in Switzerland, trying to figure out how does the immune system discriminate self from non-self. And then I got a job at the NIH. And in a lab next door to me was a guy who's now at Hopkins named Ephraim Fuchs. Ephraim was an oncologist who had just decided that rather than trying to give chemotherapy to the next tumor patient, he was actually going to try and do research and get better therapies for tumors. And he was in the lab next door. And he was asking the same questions for a different reason. He was asking, why is it that we have tumors that we know are expressing foreign proteins, either because they're virally encoded, or they're mutated self-proteins, or they were on really early in life and disappeared before the immune system ever arrived. They ought to look foreign, and tumors ought to get rejected. But they don't. And by that time, I was in old fogey mode, and I patted him on the head and said, there, as we all know that tumors are immunosuppressive. And unlike me, he wouldn't let go. So we spent the next 2 years arguing about what kind of immune system would be evolutionarily useful. What kind of an immune system would actually work and incorporate all these things that didn't fit with the self-non-self model. And after 2 years, we came up with the idea that, finding everything in the air you breathe, or everything in the food you eat, or your fetuses, or the sperm that gets you those fetuses is ego. What you need to do is fight things that are dangerous. So that took 2 years. That's all very well. We're going to say, okay, the immune system fights things that are dangerous. What's dangerous? How do you define danger in such a way that it explains what the immune system gets right and what it gets wrong, right? You need a definition that works. I mean, tumors are dangerous, they kill you, but the immune system doesn't respond to them, right? Transplants help you, but the immune system does respond to them. So, figuring out what was dangerous wasn't so easy. And it actually happened to me in the bath. And I really understand Archimedes, because I jumped out of the bath, started thinking and ran around the house. Going, I got it, which is exactly what you're making. So here it is. It's really simple. I don't know why it took so long. Something is dangerous if it does damage, and it's not dangerous if it doesn't do damage. Right? The virus that comes in and makes 3 copies of itself and leaves, you don't need to make an immune response. Right? Ephraim says you might want to welcome it because it could bring you genes you wouldn't get otherwise. So, something is dangerous if it does damage. It's really, simple. Yeah. So that was now the 3rd year into our arguments, and then we had the final question. We thought the final question, which is, okay, how does the immune system know that something has entered the body and is doing damage? And that took another year. And that happened while I was training my dog to herd sheep. Because here's what happened. It was summer. Sheep have these woolly coats. You can't train for too long, right? Because it's hot. So we train for about 10 minutes at a time, and then we rest. And we were resting, and the sheep had gone down the bottom of this little hill, and there were some woods behind, a little stream. And my dog and I were sitting up on the hill. She was kind of curled up, relaxed. And all of a sudden, a sheep came running out of the woods. Bang! Actually, they don't bang. They bang! And my dog shot into the woods. And I realized that there is a cell in the body that is just like a sleeping sheepdog. And that the alarm for that sleeping sheepdog wasn't hearing something in the woods. It was hearing the sheep. The sheep was telling her there was something wrong, right? So the idea then was that damaged cells send alarm signals. And the alarm signals are picked up by special cells called dendritic cells. That isn't fantasy, that's fact. There are these cells called dendritic cells. They are called dendritic cells because they have very long dendrites, just like neurons. They live in every tissue that's ever been looked at. So there may be some they're not in, but so far every tissue we've looked at has dendritic cells. Quiet, asleep, nose under their tails, in contact with a whole bunch of tissue cells. So in the skin, they're called Langerhans cells after the guy who found them. And each one is in contact with between 50 and 500 skin cells. And they're just quiet.
What was the name of that cell?
The Langerhans cell.
Okay.
Good.
Isn't there something similar in the pancreas?
Everywhere. There are dendritic cells in the pancreas. Oh, you're thinking of the islets of Langerhans. Right. Langerhans was a microscopist and a pathologist. He looked at lots of tissues.
Okay.
So he found, yeah, good question. So he found the islets, which are the cells that make insulin, which you destroy in diabetes, type 1. And he also found these Langerhans cells in the skin.
Okay, thank you.
Yeah, good. It takes a brave person to ask the first question. It also takes a brave person to ask the second. So, after that, it's easy. So these cells are in all tissues, they're asleep. And what we suggested is that they are the ones who pick up the alarm signals. Now, I want to backtrack a little bit and go through about 70 years of immunological theory in one diagram to show you what previous theories we're standing on and what previous theories we disagree with. Because you will continue to hear about these other things, you'll read about these other things, and I don't want you to think that I don't agree with them. And there were some wonderful thinkers in the immune system. So let's start with 1954. 1954, there was a guy named Burnett in Australia. And he was trying to understand how the immune system can fight all those different kinds of pathogens out there, bacteria, viruses, fungus, all kinds of stuff, worms, and not kill yourself. And also, how was it that when you made an immune response to, say, measles, you would now make a better and faster immune response to measles the second time? And he came up with something called the clonal selection model of immunity. And if you look at that model in modern terms, you would say that the B cells, they're called B because they're made in the bone marrow, they make antibodies. Does everybody know does anybody not know what an antibody is? Okay. They make antibodies. And his suggestion was that each B cell has on its surface multiple copies of the single antibody that it's going to make later. And that should, let's say this is anti-flu, right? So flu's in the bloodstream, there's a B cell in the bloodstream, it binds via its anti-flu antibody to flu, and it gets a signal that's called stimulation or signal 1. And the reason you can respond to flu and not your own kidneys is that early in life, all the B cells against yourself are depleted. So all you have left are B cells that can see foreign things. And mice have hundreds of millions of them. They make 35 million new ones every day. Humans, you can ramp that up about 1,000 times. New B cells are being made all the time. You can see B cells exist that can see molecules that never existed in nature. They are randomly creating these receptors in such a way that they're prepared for the unexpected. And Burnett suggested that the B cells that are against you are deleted. Then there's a cell called a killer, which does not secrete things, and it has on its surface similar receptors that can see foreign things, but they don't see foreign things alone. These killers can't do anything to a flu virus that's just floating in the bloodstream. They can only kill cells. And they kill a virus-infected cell within 20 minutes of the time that the virus has entered. So the virus doesn't have time to make new viruses yet. These are the world's best microsurgeons. They can go into a tissue and kill that one infected cell before it has a chance to multiply and stop the infection. And the way they know they're looking at a cell and not a free virus in these special Schlecker molecules and put out peptides from the virus. And the receptors on the surface of the killers look at the combination of the surface molecule and the peptide. So the same idea here, kill off all the killers that can see you early, and the only ones that are left can see far. That was 1954. You didn't know about killers, so I'm giving you the modern version of this. Go ahead.
Is there the same amount of diversity in the killer cell
Yes.
population?
Yes. Yeah, it's amazing. And it's a wholly different universe of things they're looking at, because they're not looking at flu or measles or pollen or yet. They're looking at peptides slotted into the groove of these, they're called MHC. 2 alpha helices on it. It's like two hot dog in a bun. They slot these peptides in there. If you're sensitive to nickel, which I am, some people are, the nickel is sitting on one of the peptides in the MHC. If you're sensitive to poison ivy, people say you're immune to poison ivy, you never get it. Poison ivy is an immune response. If you get poison ivy, it's because you're immune to poison ivy. If you can roll in a field of poison ivy and have no problem, it's because you're not immune to poison ivy. It's the immune response to poison ivy that gives you the rash. Okay, that was 54. The idea then was, if you get flu, this B cell starts making antibodies. It makes about 2,000 molecules per second and secretes them out into the bloodstream. And it makes an army of cells doing the same thing. It takes 3 or 4 days before you have a high enough concentration of those antibodies to really deal with flu, which is why you get flu and get sick. And then after a while, you've got enough antibody and that deals with the flu. You've got killer cells, killing flu-affected cells. It also takes them 3 or 4 days to make an army and get going. Okay. In the 60s, it was discovered that when B cells see something like flu, not only do they multiply to make an army and start secreting all these antibody molecules, they start to mutate. In fact, they hypermutate. And in fact, they mutate only that part of the antibody that binds to flu, right, say, or measles. And the idea is that they're doing this because there's only 1 in a million or so that can see flu. And the chances that it can bind it really strongly are low. So what happens as they start multiplying and making an army, they mutate. And some of them are now going to bind to the flu more strongly. And some of them don't bind it anymore at all. It's just random mutation. Now that created a problem because the idea is you're self-tolerant because you removed all your autoreactive B cells early in life. But now when you get flu, they start to mutate. And one of them could now mutate to become reactive to your own kidney or liver or heart. So how could you deal with self-tolerance in the face of all this mutation? So in 1969, Peter Brecher and Mel Cohn at the Salk Institute in California came up with a kind of cute solution. They said, if you want to maintain self-tolerance, make sure that the initiation of immune response requires 2 cells. It would be a rare day when a B cell against flu and another different kind of cell against flu would both mutate to see the same cell clone. So he invented another cell, and he called it HELP. We need a helper cell, and we now know that they were right. The cell's called T helper cells, T because they grow in the fungus. They actually said, if you really want to fix it, require 3 cells. Right? It's going to be really, really, really rare that 3 cells mutate to see the same cell protein. The problem is that when the frequency of each cell is 1 in a million, requiring 3 cells means you need 10 to the 18 cells. We don't have 10 to the 18 cells, and so they settled on 2. Right? This is how immunological theory happens. And their suggestion was this, that if the B cell gets signal 1 alone, it dies. Unless it gets a second signal they call home. only if it gets that second signal does it survive to make a response. So if one of these guys mutates to C cell, and there's no help, right, it will get signal 1 and die. And so in this way, you are constantly re-tolerizing for yourself. Does that make sense? Yes?
Do the T cells also mutate and adapt?
You're good. They don't. I mean, any cell will mutate when it's divided. But they don't have the special machinery to hypermutate just that part of the receptor that sees the foreign entity.
Okay.
Yeah? However, hang on to that question, because you're a plant. I need you in the audience.
Yes, the second question.
Yeah, right.
First and second
First
question.
and second.
Yeah? Okay, so that lasted until 73. And in 1973, we're back in Australia. And this time it's a guy named Lafferty and Cunningham, Kevin Lafferty and Alistair Cunningham. Followed tradition and added another cell and another signal. And the cell they added, they called it an accessory cell. We call it an antigen-presenting cell. You don't have to read well. You kind of immunologists use this word antigen. You'll hear it, you'll read it. It comes from antibody generator. Something that goes in and will generate or induce antibodies. We now use that term a little more loosely. Anything that will induce an immune response. So they invented these cells called cells. They invent them, they brought them into the conversation. And the reason they did that is that in the late 60s, early 70s, it was becoming clear that there's a kind of species selectivity about immune responses. In the sense that we know, for example, that if you got my kidney, you'd reject it really fast, right? And people can measure that even before you get the kidney by taking some white blood cells out of your blood and mixing them with mine, irradiate mine so that you can't do anything. And then they ask how many of them respond and how strong is that response. And what was being discovered was that a human set of white blood cells will respond more strongly to those of another human than to the white blood cells of a mouse or a sheep or a dog. And a dog is the same. A dog responds more strongly to another dog than it does to a mouse, a sheep, or a human. And yet, a mouse ought to be more different from a human than another human. And so they've suggested that there ought to be some signals that are going across from cell to cell that are species specific. And they brought in this presenting cell, and they said, this guy also needs a second signal because it could also become autoreactive. And so they brought in this cell, which we knew about, called a macrophage, or an dendritic cell, which presents what it picks up. And said when the T cell gets signal 1, it dies, unless it gets a second signal that they called co-stimulation. So remember, signal 1 is stimulation. Signal 2 here is help. Signal 2 here is co-stimulation. All right, that was 73. For the next 13 years, this signal was studied. This one was totally ignored. You could get NIH grants to study the collaboration between T cells and B cells. Nobody wanted to hear about the interactions between T cells and dendritic cells or macrophages. Why? Because it doesn't fit with the model. Here's the thing. If the T cell or the B cell is the cell that controls immunity, then you can have a self-non-self model because you can remove the ones that are anti-self. The thing about macrophages and dendritic cells is they pick up anything and present it. There's some chairs here. I'm known for going over, so you don't want to stand if you don't want to.
So
Macrophages and dendritic cells are interesting cells. They live in tissues. they're asleep. If they wake up, they start drinking their environment. They drink their entire volume every hour. They throw out the water. They keep the solid bits, the proteins, carbohydrates, etc. that they find, break them up into little pieces, and display them on their surface. They pick up anything. Now, let's say you have chicken pox. Right? You get chicken pox in the skin. It induces an immune response. I'll tell you how in a minute. The local antigen-presenting cells wake up. They drink their environment. They're going to pick up pieces of chicken pox from burst cells, pieces of cells, whatever. And they're going to leave the skin and go to a local lymph gland. And there they sit. And the T cells and the B cells are passing by. They're bar hopping. They're going from lymph gland to lymph gland to lymph gland to lymph gland. And if they find a presenting cell presenting their particular molecule that they can recognize, they stop. And the activated presenting cell gives them the second signal that tells them make an army and go make an immune response. What they do is they multiply. That's why your lymph glands get bigger. There's more and more cells in there, right? 3 or 4 days, they get bigger. And then after that, they get smaller again because the cells have left. And they're going out into your bloodstream and they're going out into other tissues looking for the virus that they've been turned on to. And when they find a cell that has it, they kill it. So these guys pick up everything. So the reason people ignored this idea was that if the cell that starts an immune response is not specific but can present anything, it's going to present self and non-self. And therefore the self-non-self model couldn't work. So, doesn't fit your model, you ignore it. So, what happened was in 1986, at the NIH, Ron Schwartz and Mark Jenkins discovered co-stimulation by accident. They were trying to find exactly what the peptides were that were presented. And so they were fixing these cells with a molecule called glutaraldehyde, which basically turns them into little billiard balls. And they found that even though they knew they were presenting, they would not turn on a T cell. And so this paper by Lafferty and Cunningham had sunk so much like a stone that when Schwartz and Jenkins rediscovered it, they referenced this second signal help and not this second signal. Anyway, it was rediscovered and people started working on it. And everybody was really confused because, you know, we'd grown up with self and non-self. And now we had this cell that was starting immune responses that couldn't tell between self and non-self. So that only lasted for 3 years. In 1989, a guy named Charlie Janeway, he's dead, unfortunately. He and I used to go to meetings and scream at each other, and then he found a way to put co-stimulation back under the control of the self-non-self discrimination, a slightly different kind. He said that the antigen-presenting cells themselves have receptors, not made newly in each individual, but genetically encoded, that can recognize evolutionarily conserved molecules on very distant pathogens. So he said, bacteria are so different from vertebrates who have immune systems, that we could have genetically encoded receptors that recognize bacteria. And he called these pattern recognition receptors. And he said what they bind is conserved molecules on bacteria. Now, the most important thing he said is that the APCs are usually off. Up until that time, up until 89, people thought that the immune system was always on. The lymphocytes are constantly on, they're constantly going around, they're constantly looking for things, they're constantly fighting things, da-da-da. He said, the immune system is off, and you turn it on when one of these sleeping presenting cells binds to a conserved molecule on a bacterium, and that wakes it up. And the relevant part of waking it up is that it now will give a co-stimulatory signal. And when I first heard this, I said, Charlie, it doesn't work. You know, it tells you how you get responses to bacteria, but it doesn't tell how you react to transplant, or cure a tumor, or get autoimmune disease. And his answer was interesting. He said, oh, transplantation is a modern invention. We didn't evolve to deal with that. And tumors and autoimmunity kill you late enough in life that you've already had your kids, and so evolution doesn't care. And after a minute, I said, well, you know, wait a minute. If you want to describe what the immune system does, you have to describe what it does, not just what you happen to think it evolved to do. And it does reject transplants, and it can cure a tumor, and it does give you autoimmunity. Right? So what Ephraim and I did was we followed tradition. We added another cell and another signal. Yeah? And it turns out that this is it. There are no more cells you can add. This is the end of the line. Because what we said was every tissue, we brought every tissue into the conversation and said that healthy cells, or cells that die by programmed cell death. So a cell that dies, so cells die a lot, right? The cells in the bone marrow that die all the time, the cells in your skin that die all the time, the cells in your intestines that die all the time, making room for new cells, etc., right? And they die by something called programmed cell death. Except for the ones in the skin and the gut, which are just sloughed off into the environment. Most of the cells that die in the body by programmed cell death are scavenged either by their neighbors or by special scavenger cells. And so what we said was a cell that is healthy or dies by programmed cell death and is scavenged by its neighbors is ignored by the immune system. But a cell that is stressed, injured, or dies badly sends an alarm signal that's picked up by the presenter. In fact, we're suggesting that these receptors that Charlie found are really there to look at self. My favorite, there are lots of alarm signals. Now, I published this in '94, sank like a stone. In '99, we published the first test of it, killing cells by apoptotic death and by necrotic death, and injecting them and asking if the mouse would make an immune response. And they do if it's the necrotic or bad death, and not if it's good death. And then people got excited. And slowly over the years, people have been finding the alarm signals. And it's interesting. Anything that is normally found inside a cell and not outside a cell can act as an alarm signal. All you need is a receptor on the APC that has formed. My favorite alarm signal was discovered after Charlie died, unfortunately, and it's mitochondria. So mitochondria are these little powerhouses inside cells, and they're really bacteria. Long, long time ago in evolution, these bacteria got inside a cell and stayed there. And without them, we don't survive. But what's interesting is they still look like bacteria to the immune system. So if you get major trauma, you get lots of mitochondria released, and sometimes you can get an immune response, which then kills you because it's too strong. There are lots. DNA is an alarm signal, RNA is an alarm signal, heat shock proteins are alarm signals. A molecule called hyaluron, which lines your blood vessels. If you bruise it, the breakdown products are alarm signals. All of these molecules will wake up and antigen present itself. So it doesn't really matter what kind of injury you have, whether it's a heat shock, a cold shock, a virus, a bacterium, a fungus, cut from a surgeon, you get an alarm signal. All right, so we followed tradition. We added another cell. We added another signal. We said that without the alarm signal, you don't get an immune response. We just took one more step down the same path as everybody else. But it turns out that if you take that step with me, you fall off a cliff. And now you stand at a different place. And you look at the immune system from a different point of view. And it turns out that if you look at it from that point of view, you can explain almost everything that it does right and almost everything that it gets wrong. So here's the list. We're not going to go through the whole thing, and we'll be here till midnight. You can explain why you don't heal yourself at puberty. Or at least, a car wreck rather than immunologically. You can explain why lactation explain why mothers do usually reject transplants, and you don't usually reject tumors. You can explain why you get autoimmunity. You can explain why there are parasites that live in intimate contact with the cells of the immune system, like the filaria worm, which gives people elephantiasis. It lives in the lymphatics. In the lymphatics is where the immune system cells, the white blood cells, traffic, and yet you don't get an immune response. You can explain something called graft versus host disease, which is when you give a bone marrow transplant to somebody who's been irradiated, the cells in the bone marrow kill the patient. And what you can't explain, at least with the original danger model, was allergy and asthma. And that didn't bother me. And the reason it didn't bother me is that allergy is a problem in a different universe of question. So what do I mean by that? So when the immune system is faced with something, the first question it has to ask is, do I respond or not? Right? The old model said you responded if what you were facing was foreign. Danger model says you respond if what you're facing is doing damage. Once you decide to respond, you've got a second question to ask. What kind of response do I make? Right? Do I send killers to catch a virus? A special kind of antibody called IgE to catch a worm? How do I know what kind of immune response, what kind of weapons to turn on? And I think that's the problem in allergy. If you look at allergens, there's only a few proteins in most individuals that are allergens. So for example, the house dust mite probably has about 25,000 different proteins. 2 of them are allergens, derpy 1 and derpy 2. The cat has a lot of proteins. 1 allergen, FeLD1, 1 main allergen. So people in allergy have been asking the question for a long time, what makes an allergen an allergen? And my answer is, an allergen is an immunogen. An allergen is something that either causes damage or is packaged with something that causes damage, like venom, or mimics one of the alarm signals. Or you get it when you're damaged. So this is something I just learned 2 weeks ago when I was at Master University, and there's a guy there who works on peanut allergy. And it turns out that there was a rash of peanut allergy in the 50s or 60s, I think it was the 60s, that then went away, and now there's another one. Well, that rash of peanut allergies in the 60s turned out to be due to baby oil that mothers would put on their babies when they had diaper rash. Diaper rash is dangerous. Yeah, it's so dangerous. The skin is hot and raw, and yeah, your Langerhans cells are active, and they were putting oil, and peanut oil is a really nice oil. When they took peanut oil out of the baby creams, that particular rash of peanut allergy went away. So it'd be interesting to see where it's coming from now. So that's packaged with something, or being given at a time of danger. What the danger model cannot answer is, why do some people make IgE and get allergic and other people don't? So I've been working on that. Ask me back in a year or 2. Okay, so the basic assumption. Here's the basic assumption. Let me give it to you again, okay? You never, I tell my postdocs that if you say never or ever or always or none, in biology it's a lie. Right? Because there's always an exception. But I'm going to say it anyway. You don't get a primary immune response until you activate a presenting cell. And you activate a presenting cell with an alarm signal from a damaged tissue. Right, so, pretty simple. Got it? Can you hold on to that long enough for the rest of the lecture? Mm-hmm. Okay.
But I have a question about the, so
go ahead.
Is there a difference between, say, acute damage and like chronic, long-term, subtle damage that accumulates over time?
Probably. There's going to be a difference in the cell that does it. There's going to be a difference. So some alarm signals are constitutive. They're just there in the cell, DNA, RNA, mitochondria, right? Some alarm signals are actually produced. So a cell that's virus infected, for example, knows it's virus infected, and it makes interferon alpha. And that stops the neighboring cells from getting infected. It also calls in the immune system. It activates the local presenting cells. So there can be acute damage. There can be acute subthreshold damage as well. So I think that it takes a certain amount of, so we have evidence that it takes at least 2 dead cells worth of alarm signals to turn on a presenting cell.
I'm thinking of things like arthritis and, you know, where there's tissue damage, mechanical tissue damage over longer periods of time that, you know, is from a chronic standpoint.
If you guys are willing to stay here, I'll come back to that.
Okay.
Yeah.
Sure.
That goes under autoimmunity, and I need to tell you a lot more things before you're ready to hear. I have to warn you, you're going to have to eventually vote with your feet. I was once invited to a student's retreat in France and given an hour talk, and I ended up talking for 2 hours and 40 minutes, and they videotaped. So, okay, I won't do that, I promise you.
So, do you think that you have to have tissue damage? You can't have just like a PAMP or something like LPS that finds TLP?
So LPS is interesting. I brought a slide, no, I think I forgot it actually. LPS and cardiolipin. Cardiolipin is a molecule found in the inner membrane of mitochondria. Look awfully similar. But the thing about LPS, so I think LPS is actually a bacterial alarm signal. So LPS, for those of you who don't know, lipid polysaccharide is a molecule from E. coli and other gram-negative bacteria that the immune system responds to. Some mice respond really well to it, humans less well. There's a kind of species specificness about this. And if you look at the molecule itself, it sits in the outer membrane. So these bacteria have 2 membranes. Called gram-negative because they have 2 membranes. This gram stain can't go in. And the LPS molecule is a lipid with a polysaccharide. And it's the lipid part which is immunostimulatory. And if you look at that lipid part in living happy bacteria, it's hidden in the membrane. So Song Song was a Korean postdoc, arrived in my lab and on day 1 told me that Charlie Jamie and I were both wrong. Actually studied LPS. And he said, you know, it's not exposed on living bacteria. And if you look at the experiments people do, they use heat-killed bacteria. Yeah? And he said, you know, maybe it's an alarm signal for bacteria. Maybe this is one of the alarm signals that our immune system has piggybacked on because bacteria are evolutionarily so much older than we are. He says bacteria, you know, we think of bacteria as these single cells, but really they live in colonies. And the colonies they live in are usually their daughters and mothers. You know, they divide and make more of themselves, so they're genetically fairly identical. And so you would want to know if your colony mates were dying. Because then you could either upregulate a different nutrient handling system, or you could leave, or you could sporulate, or, you know, do something. Right? So he thinks LPS is an alarm signal for bacteria.
Like
Form sensing for death, yeah, or destruction of something, yeah. Yeah, there are form sensors that just tell you how many of your friends are around. He thinks it's one that tells you how many of your soulmates are dying. There are a lot of like that. The receptors that are known to bind to LPS or lipid-teichoic acid and so on have also, as people studied them more and more, been found to bind to endogenous alarm signals. And so we think that that part of immunity is not new. It didn't evolve with us. It got invented long, long ago, and we're just using it.
How would a transplant an organ, like if I gave somebody an organ, how would that stimulate an antigen presenting cell?
We're going to get there. We're going to get there. All right, so here's a good question. Here's what we're going to do. We're going to take these 3 foreign tissues, right? You're behind the post.
Yeah.
Fetuses, surgical transplants, and tumors. All right, I'll look at them. Fetuses, surgical transplants, and tumors. I'm going to use those to illustrate how the danger model explains things. And the rest just have to come some other time. Right? So I said, Dr. Mom can explain all this stuff. And why an allergen is an allergen, but not why some people do respond and others not. All right, so let's start with fetuses. So fetuses were actually the first problem self-harm had. So Peter Medawar won a Nobel Prize for doing an experiment putting adult white blood cells into baby mice and showing that when those baby mice grew up, they would accept a skin graft from the donor of those white blood cells. So that's where people got the idea that you learn self early in life. You couldn't do it later. We've now done it later. But he realized that fetuses are a problem because they weren't around when you were learning self. Mothers ought to reject fetuses. And in his inimitable Oxford-educated British arrogance, it never occurred to him that the model might be wrong. He suggested instead that there was something wrong with female physiology. He said fetuses immunosuppress their mothers. Now, that's an evolutionarily really smart idea. Right? You take the individuals that are carrying the next generation and you turn them into AIDS patients. So I've learned in my career that if something seems evolutionarily stupid, it's probably wrong. I lost my blue markers. And it turns out it's wrong. Good, thank you. How many of you carry fetuses? Did you have to live in a boat? No. Has anybody noticed? Right? Never had kids? Yeah, all those cows and sheep out there, you know, having lambs and cows and whatever. They live in boats. I mean, there are some parasites that take advantage of the pregnant state. But pregnant individuals are usually pretty healthy. So, it's wrong. Evolutionarily found wrong. And so he suggested a second idea, which is also written in textbooks. By the way, that one is still in textbooks. That the fetus immunosuppresses the mother. Here's the second one, that the placenta acts as a physical and immunological barrier between mother and fetus. All right, that's great. That means if your fetus gets infected, you can't do anything about it and you're dead. Also wrong. It turns out there's traffic across the placenta in both directions. And so there are people in California who, well, it's now spreading around the country, who can isolate from the blood of a pregnant woman the fetal cells that are circulating there, and they can isolate them and do DNA analysis to do a non-invasive prenatal diagnosis. So there's traffic from fetus to mother. There's also traffic from mother to fetus, and this was first discovered in Leiden, in the Netherlands, by Jan van Vogt, who used to run Eurotransplant. And they have people waiting for kidneys. And when you have a kidney recipient, you know, they're on dialysis, they need a kidney, and you have a living donor, you always test the immune response of the recipient against the donor before you do the transplant. Because if it's already strong, it's a waste of a transplant, it'll get rejected, even if you give the immunosuppressive drugs. So they have this whole list of people that had living potential donors. So they tested them. Now, mothers are often donors. And it turns out that 25% of the people waiting for a kidney and tested against their mothers did not make an immune response against their mother's tissues. They made an immune response to their fathers, you know, the non-inherited part of their fathers, but not the non-inherited antigens of their mothers. Why? So Lee Zhang and Rick Miller followed this up in Canada with mice, where they could genetically mark father, mother, and progeny mice. And they discovered that 25% of adult mice are microchimeric, meaning they carry small numbers of white blood cells from their mothers. 50% of human skids, these are the boys that do have to live in a bubble, are carrying T cells from their mothers. So there's traffic from the fetus to the mother, and there's traffic from the mother to the fetus, and yet fetuses are usually not rejected. Why not? Right, so the danger model says it's because they don't look dangerous. Now, what's important here is they don't look dangerous to the immune system, right? Because the death that goes on in fetal life is programmed silent. So, you know, you get this primitive bump that's going to turn into an arm and eventually into a hand. It turns into a hand because the cells between the fingers die, and they're scavenged, and they don't release alarm signals. So as long as the fetus is healthy, and the death that's going on during that development is programmed cell death, it's not releasing alarm signals, and so it doesn't turn on an immune response. Now, if it gets infected, it will turn on an immune response, and the mother will fight that infection. And sometimes in the process, she clears the fetus along with the infection. And in fact, we know from the medical literature that spontaneous miscarriage is a common concomitant of infection in pregnancy. So, danger model says you don't reject the fetus, not because it immunosuppresses the mother, but because it doesn't look dangerous. Now, it is dangerous. It saps calcium out of your bones, and in starving populations, the very young, the very old, and the pregnant females are the first ones to die. But it doesn't look dangerous to the immune system. Right? Now, I'm willing to wager there's at least one person in the audience wondering, what about RH disease? Anybody? Bring it up. Oh, you weren't here when I said you have to speak during the...
I was wondering about anticardiolitis and
Yeah.
spontaneous abortion and lupus.
Lupus, we'll have to do lupus where you are. Stick around if you want. We'll do RH disease.
Yeah, it has to be primed at one time.
Yeah. So RH negative mothers that have RH positive babies, this is an antigen on red blood cells, have a problem. They get immunized. They make antibodies to RH. They end up with blue babies. Now, God is in the details. The detail that's important here is that it's a problem in the second pregnancy and not the first. You get primed at birth. Birth is dangerous. You have ripping and bleeding, surgeons with knives. It's a dangerous period. And in fact, physicians know this. And so what they do is a few days before this woman is going to deliver, they pump her full of antibodies to RH to hide it from the immune system. And then she's usually okay. Occasionally, you get a woman who starts making anti-RH antibodies towards the end of pregnancy, first pregnancy. And Liz Bonney, who was an OB-GYN resident in my lab for a while, says that there are women that have what are known as fragile placentas. So the fetus puts these little fingers of tissue into lakes of maternal blood. And every so often, you get one that they're not very strong, they break off. And that is not programmed cell death. And I'm predicting that those women who get RH antibodies towards the end of first pregnancy, if you could measure it, you discover they also have this fragile placenta syndrome. Usually it's the second. Okay, so you don't reject the fetus because it doesn't look dangerous. Where is the danger in a transplant?
I guess from the scalpel, because of
The surgeon, absolutely. Surgeons are dangerous. Yeah. They cut, they burn small vessels, they put organs on helicopters and fly them around the country. They get ischemia and post-reperfusion ischemia, which means that they put these organs in a special bath with nutrients and cool it. By the way, that's a heat shock. And while they're transporting it from one county to another for the right recipient, it's not getting a lot of oxygen. And so what happens is it upregulates oxygen handling enzymes. And then you put it in the blood, you bring it back up into the body, you bring it back up to 37 degrees, and you let the blood go in, and now all of a sudden there's all this oxygen. And all these oxygen handling enzymes, and they make oxygen radicals. So you get a whole bunch of oxygen radicals that do damage. It's called post-reperfusions. So there's a lot of non-programmed cell death involved with a transplant. So the prediction from the danger model is that if you could do a transplant without doing damage, it wouldn't be rejected. But you can't. Now, even taking blood out of one person and putting it into the next person, you cool it. Yeah? It's a heat shock. You look at dendritic cells cross-eyed and they activate. And I tell you, I know this. We grow them. We take bone marrow from mice and we put them in dishes and grow them for 6 days in certain hormones that tell cells to become dendritic cells. And then on day 6 we use them. If day 6 is a Friday, that population of dendritic cells has a greater frequency of spontaneously active. It's because the incubators don't get opened as often on the weekends. When you open the incubator, you know, you change the temperature a little, you change the gas a little, you change the pH. So look at them cross-eyed. Alright, so prediction is, if you could do a transplant without brain damage, it wouldn't get rejected. You can't do it, but a guy named Colin Anderson, who was still in my lab, now he resigned in Canada, did the following experiment. He asked the question, if an immune system sees a foreign transplant in the absence of damage, would it reject it? The way he did that was to take skin mice. These are severe common immunodeficiency. These are mice that have to live alone because they don't have T cell diseases. They can't reject transplants. And under the kidney capsule, he put a baby heart. It had to be a baby heart, that won't fit. And it's not rejected because these mice don't have an immune system to reject them. He left that heart sit for 9 months to heal. Now, why 9 months? Well, you couldn't leave it for 2 years because you only have 2 years post-op at the NIH. You got to do the experiment more than once, so people won't believe you, right? And he'd already discovered that with a skin graft, doing the same thing with skin, that at 5 months, the skin still hadn't completely healed.
And so what?
Still had not completely healed. Right? By quantitative PCR, you could see repair and remodeling enzymes still up. And we just between 7 VEGF and so on. So he left a free guest, left it for 9 months to heal. And at that point, he gave the mouse back an immune system by putting in precursors for T cells and B cells added. And he asked, when those cells grow up and recognize that foreign transplant, will they reject it? And the answer is no. Now, if you'd only left it for 1 month, it was rejected. If you left it for 9 months, it was not rejected. 1 month is not enough. I now think, I have no evidence for this, it's total handwriting. By the way, people ask me sometimes if my wrists don't get tired, yeah. I now think that the tissue doesn't become fully healed until it becomes re-innervated. And I would love to test that, but I don't know. So, anyway, 9 months not rejected. So then he tested a second aspect of the danger model. So the danger model, remember, says that if you have a tissue which is happy and healthy, and its antigen-presenting cells are asleep, and it's presenting a window display of what's in it, in the surface, and these guys are picking it up even though they're asleep and also presenting it. That if a T cell seeing this will get signal 1 and not signal 2 and die. Danger model says that your body is constantly tolerizing for itself. As you make new T cells in the thymus, new B cells in the bone marrow, as they come out, a T cell that comes out of the thymus that is able to attack the liver. If your liver is healthy, will eventually go through the liver, get signal 1 without signal 2 and die. So you're constantly inducing tolerance to yourself. So he tested that. He allowed the immune system and this unrejected heart to interact with each other for another 5 months. And to ask if the animal had become tolerant, he immunized it. He vaccinated it against the donor. And he tested killers. Now people say I never show data. I'm actually going to show you one piece of data. Oops. because they didn't get a heart. They're litter mates, they sit in the same environment for 9 months, and then another 5 months, and then we immunize them. And we tested them for killing. What we do is we increase the number of killers, and we look for an increase in kill. And you see that 2 of these mice killed phenomenally well. It takes very few of them to get to maximum kill of targets, donor cells, and 1 killed very well. And they don't kill cells. These are the ones that had the heart for 1 month. They didn't even need to be immunized. They also kill, not quite as well. These are the 3 mice that had their hearts for 9 months before they got our immune system. They didn't reject, and if you left them for another 5 months, they couldn't be immunized to kill. So what does that mean? It means that A, if the tissue is not sending alarm signals, it's not going to be rejected. And B, if you leave it long enough, it will induce tolerance for itself. So, when I gave these data to the annual meeting of the American Society of Transplant Surgeons, the surgeon got up in the back of the room, stood up, pointed, and said, "You're wrong, dearie." He said, "I have kidney patients that have had their kidneys for 15 years. And some of them get sick of the effects of cyclosporin, which is the main drug that they were using as an immunosuppressant. And they stop taking their meds. And depending on what center they're in, 50 to 90% of them now reject their kidneys. Now, 15 years ought to be long enough for a kidney to tolerate it. So, luckily I'm a young age, we had lots of seminars, and that was at a period when cyclosporin was all the rage, and so we had lots of seminars about how cyclosporin works. Well, it turns out that cyclosporin blocks signal 1. Right? It blocks the calcium flux, which is the result of a T cell or a B cell sequence. And we're saying in the danger model that in order for a T cell to become tolerant, it needs to see its antigen and get signal 1 without signal 2. If it can't get signal 1, it can't become tolerant. What happens is cyclosporin basically blindfolds them. Yeah? And so for 15 years, they're walking around. Right? Blindfolded. And then you take the drug away. And they're fine until they get an infection. So for a while, I was getting emails from a woman who was a heart transplant patient who had gone off her meds. Now, this is a brave thing to do. You lose your kidneys, you're back on dialysis. Lose your heart. So she stopped her meds, and she was good for 18 months. And then she got a recrudescence of a virus called Epstein-Barr virus, which 95% of us carry, and started to reject her heart. So they put her back on her meds. 18 months. No problem. All right, so the reason you don't reject the fetus is it doesn't look dangerous. The reason you reject transplants is they do look dangerous. You've got to figure out what the alarm signals are. There's a lot of them. We'll never be able to block them all. But what we can do is block signal 2. And there's a guy named Alan Kirk. Believe it or not, he's Commander Kirk. He's in the Navy, or at least he was in the Navy. He's a transplant surgeon. And he transplanted kidneys in monkeys and blocked signal 2. Now, remember, what we're saying is you induce tolerance by allowing the cell to get signal 1 without signal 2. So he blocked one of the signal 2s for 5 months and then stopped. And none of his monkeys rejected. It was in Nature Medicine about 8 years ago. Those monkeys are still good. None of them have rejected. One died. It's sad. You know, one died. They bleed them every so often to test them. And they bled one of them and he dropped dead. But when they checked his kidney, he was fine.
How did they block signal 2?
How did they what?
How did they block signal 2?
With an antibody to, all right, so there are 3 very well-known signal 2s. One of them is a molecule on the antigen-presenting cell called CD40. And on the T cell is something called CD40 ligand. And he blocked that with an antibody. He still got antibodies to the grafts, and he wanted to do a clinical trial where he also blocked the other two signal twos that we know of, which are called B7-1 and B7-2. And here's how medicine works these days. Chiron owns the antibodies to one of them. Merck, I think, owns the antibodies to the other one, and a third company owns anti-CD40. And none of those companies were willing to be part of the clinical trial that also depended on reagents from the other. So Kirk is now making his own. He's at Emory now. No longer Commander Kirk. He's a star. You can follow him if you're interested in transplants. He's a star. Okay. So you don't reject a fetus because it doesn't look dangerous. You reject a transplant because it does look dangerous. Why don't you reject a transplant? They're dangerous. They kill us.
They like encased in something?
Some not. Leukemia is they're in contact with the immune system.
They grow there without inducing any kind of cell death around them?
They, at least in the beginning, what's wrong with the tumor cell is it's too healthy. Right? It's lost growth control. That's what's wrong with the tumor cell. Right? Cells, the growth of cells is under tight control. Your skin is growing outwards, but it isn't growing inwards, right? The melanocytes in it that make you colored are sitting there. Sometimes they have to replace themselves, and if you take out a big piece of skin and new skin grows there, the melanocytes around the area have to make more in order to fill in that area. But then they stop. You can cut out 90% of somebody's liver, and that liver will now regenerate itself. When it gets back to being a normal liver size, it stops. The tumors have lost the stop. So what's wrong with the tumor cell is it's just multiplying or dividing. Since multiplication and division in biology are the same. Yeah. So it's just making more of itself. And at least in the beginning, is it sending alarm signals? And suppose that it actually is expressing some tumor molecule, some mutated molecule or viral molecule or something that's in that cell. What's going to happen to the T cell that can see it?
It's going to self-destruct.
Yeah, it's going to get signal 1 without signal 2 and die. So as the tumor is growing, it's inducing tolerance to itself. That's, why they grow. Now, it used to be thought that you got a tumor every 6 minutes and the immune system was on surveillance mode. It's called surveillance mode. And it killed off most of those tumors, and the few tumors that actually grew out were the ones that escaped the immune system. And then when I was a graduate student, somebody discovered a mouse called the noob mouse, which has no hair, also has no thymus, therefore no T cells. And it doesn't have more tumors than normal mice. And then people made the rag knockout mouse, which has no T cells and no B cells, and it doesn't have more tumors than normal mice, under normal conditions. You can get it to make tumors by putting a little piece of methylcalanthine under its skin, which is a carcinogen. So the idea that the immune system was constantly controlling for tumors kind of died.
Don't immunosuppress people at a high rate of tumors up?
They do. They absolutely do. And some people think that that's because they're immunosuppressed and no longer surveying for tumors. And some people think it's because now you allow viruses that are tumorigenic to come out, whereas normally the immune system would be keeping them down. And some people think it's by whatever you're doing to immunosuppress them. I mean, irradiation, for example, can create tumors. So the danger model says don't reject tumors because tumors don't look dangerous. Now, the thing is that after a while they will. Especially if it's a solid tumor and it's poorly vascularized, doesn't have a good blood system, it'll start to die in the middle. Now, starvation death is also programmed cell is not going to get you an immune response. But eventually, when this tumor gets to be that big, and you get enough death in the middle, you will get enough death to overload the local scavenging capacity. And now, even though the cells die by programmed cell deaths, they can't get scavenged. So they're dead, they'll fall apart. It's called secondary necrosis. And there they release alarm signals. And now, you will get an anti-tumor response. And how many T cells do you have left that can see it? 3. Right? The ones that came out of the thymus last week. And they will, if it's a tumor that allows antigen-presenting cells to go in there and wakes them up by alarm signals, those antigen-presenting cells will go to a local lymph gland, and they'll sit there expressing the tumor antigens they found, right? And then 3 T cells, that 1 in whatever gazillions that you have, will eventually get there. And it'll get turned on. And it'll make 30 killers. That's it. On average, about 30. And each one of those killers can kill 3 to about 30 targets once it's been loaded. So they get activated, it takes a few days. Think of it as loading the gun. They go out, they kill their targets. Now they need to be reloaded. Takes not quite as long the second time, but it still takes a couple days. They go back to the drain lymph node, go back to the tumor. What's happening while these Q cells are killing their 3 to 30 targets? The tumor's making millions more. Right? You've lost the race. Now there's a guy at the NIH named Steve Rosenberg, who's America's Mr. Cancer. And he had kind of brilliant idea, long time ago. He said, let's take what he called the tumor-infiltrating lymphocytes. So the cells that are in there valiantly trying to get rid of this tumor. Let's take them out, grow them up in bathtubs full of a molecule called IL-2, which is a growth hormone for killers, and put them back in. Right? Expand them and put them back in. And if you've seen Steve give a talk 10 or 12 years ago, he had a set of slides he always used. It was a guy who had a metastasis of a melanoma, skin cancer, in the lung, the size of a softball. And he pulled out pills from a skin lesion, threw them out, put them back in, and the tumor went down to the size of a baseball. He did it again. The tumor went down to the size of a golf ball. And then he stopped. And the tumor grew out and killed the patient. Why did he stop? Good question. I've been asked over that for 13 years. Keep going, keep going. Yeah, exactly. Keep going. No. And he's not the only one. It's amazing to me. So the transplant community is pretty much on board. Yeah, not everybody, but most of them, right? Danger, and they're working on doing less damage and less danger, and when they do bone marrow transplants, they're doing less damage to the recipient and so on. The tumor community, mostly ignoring it. Right? Now, Rosenberg's not the only one. They're in vaccine mode. I mean, tumor immunologists want to make a vaccine that works in 2 shots.
The vaccines are for all speak. They're just for everyone, so why vaccinate?
Oh, vaccinate the ones you've got it to.
Oh.
Right?
Yeah.
So you've got a melanoma, we take the melanoma antigens, we make a vaccine of it, we start injecting you. We inject you twice. Why? Because they're in vaccine mode. Their blinker is the vaccine mode. Because a prime and a boost works for viruses and bacteria. Why does it work? Because you give the vaccine, your immune system, and the adjuvant, right? The immune system thinks you've got an infection. Those few 1 in a million cells now becomes 100 or 1,000, whatever. You increase the number so that next time you're ready to go. You give a boost, that increases the number some more. And now they go into a resting memory state. And they circulate as resting memory cells. And when the virus comes back, it does the damage that initiates that memory response. But tumors don't do that. And if you vaccinate and get killers that will kill tumor cells. What's interesting is, you know, the godfather giving the gun to one of his men and saying, you know what to do with it, right? The killer comes up to a tumor cell and tells it to die. And it dies by programmed cell death. The immune system does not throw fuel on a fire. The killing done by a killer cell does not release alarm cells. You need real damage to do that. So you will see in the Journal of the American Medical Association, the New England Journal of Medicine, the Journal of Experimental Medicine, you know, whatever medical research journal you read, every couple months, somebody trying to make a better tumor vaccine. People at Hopkins trying to make a better tumor vaccine. A lot of them. And I think they're right and they're wrong. There are a lot of tumor vaccines out there already. And they're not working. And I think they're not working because they're bad vaccines. I think they're not working because they're not being used properly. And they're not being used properly because people are basing their use on the wrong assumption. This is really a case of how what you think influences what you do. Right? So their assumption is, it's not self-control, that once you turn the immune system on to some entity, the mere presence of that entity will maintain the response. And I say no. You activate an APC, it lives for, depending on which experiment you look at, 3 to 21 days. And then it's dead. The activated APC does not stay activated forever. That would be really dangerous. You'd get autoimmunity all over the place, right? You need to activate new ones. And so you see these studies where somebody has a tumor growing, it's growing, and they do something. You know, they vaccinate because they know some of the tumor antigens, or they inject bacteria into the tumor, or bacterial products. And what they get is, you know, at the time they vaccinate, the immune response comes up, it comes down. Killers are gone into a memory state by about day 14. So at the peak of that immune response, you're going to be killing tumor cells. And if you've gotten rid of the last tumor cell, great. But if you haven't, your immune response is coming down, the tumor will start to grow again. So that's the prime. Then people boost. And you know, it comes down again. You get another immune response. And then they stop. So Steve Rosenberg gave a talk at the Center for Human Immunology at the NIH about 8 months ago now, where he happened to let slip that he had treated 2 patients with TILs 3 times, and he cured both of them. Now, cure is not a word that loosely passes Rosenberg's lips. No. For him to be cure means the guy's lived 5 to 8 years without the tumor coming back. So after his talk, I asked him, why don't you do that more often, right? Why do you stop? And his answer was interesting. It was truly interesting. So he said, I'm 24 hours in a day. I only have so much funding. I only have so many clinical fellows. And I only have so many people that are tumor patients that are willing to give their bodies to science. I want something that's going to cure tumors in 2 shots. And I say to him, ain't going to happen. It's not going to happen. So there's one guy that listened. I'm going to give you this one last experiment. You're still here. You're amazing. One last experiment, and then we'll stop. There was a tumor vaccine that originated in California at Stanford that was pretty astonishing when it first came out. And then it turned out to not be as good as people thought it was going to be. But a guy named Ron Levy at Stanford is working with people that have lymphoma. Now, lymphoma is a B-cell tumor. And what happens is one B-cell, for some reason, people have lots of ideas, starts to grow. And it continues to make the antibody it was making. But it now fills up your blood. And what they did is, from each individual patient, they took the antibodies that that person's lymphoma was making, isolated them, and made a vaccine of it. This is now individualized tumor vaccine. And they started injecting, they attached the antibody to this protein from deep sea keyhole limpets. So keyhole limpet is, like those things that live on the sides of boats. And their blood's blue. And it's called keyhole limpet hemocyanin. It's huge, it's a million molecular, it's a huge molecule. They use that as an adjuvant. They attach it to the antibody and they started injecting these people. And their protocol was 5 injections. So they're doing better than a lot of people. And their phase 1 clinical trial was astonishing. So phase 1 is a safety trial. You don't expect it to work, right? You have people that are going to be dead in 6 months. They failed everything else that can be done for them. They're donating their bodies to you, and you just want to know if you're going to kill them any faster than they would have died. Okay? Phase 1, they had 6 patients. 4 of them went into remission. Unheard of. Right? These are people who had failed, they had chemo, they had whatever, they failed, they were dead, they were on their way out. 4 of them went into remission. 1 guy died. And at the end of the trial, 1 guy had what they call minimal residual disease. They could still find a few cells. And I asked the woman who was running the trial, what are you doing? She said, we're waiting. I said, why? She said, well, we've killed 99.9999% of this tumor. The cells that are left must either be escapees or immunosuppressants. And I said, no, they're just not presenting cells. Boost him. And they wouldn't. Now, I understand it's a clinical trial, you have to plan them, you have to get IRB approval, and you can't change in the middle of the trial, right? But that was a long time ago. And they still don't. And Rosenberg won't even keep boosting with mice. It's amazing. I know we have spent around the world gazillions of dollars and francs and euros and rubles and God knows what, trying to cure cancer, right, since Nixon declared war on cancer in 1971. And we're doing a little better than we used to do. But still today, a diagnosis of cancer is pretty much a death sentence. So, Levy had a postdoc named Larry Quack. And Larry Quack had a postdoc named Maurizio Bendanti. And Bendanti works in Pamplona, Spain. And I was in Spain talking many years ago. I've been saying, by the way, I've been saying this for, I don't know, 15 years. And Bendanti listened. And he decided to try just continually boosting. It took him 4 years to get IRB approval. People were worried about autoimmunity and all kinds of other things. Right? So what did he do? He published this paper in the Journal of the National Cancer Institute. It got a big news and views from a guy named Dan Marlowe. He said, I don't know why he did this, but it seemed to work. He took a bunch of patients that had lymphoma, had been given chemotherapy, had been in remission for a while, and then relapsed. Now, what's known in lymphoma is once you relapse, meaning you get the tumor again, they give you the chemotherapy again, you're going to relapse again faster. And the reason is you relapse because there were a few cells that were kind of resistant to the drug. And of course, now they're the ones that have grown out. And out of that few cells that were resistant, you now have a whole bunch, and by mutations, some of them will be more resistant. Right? So what happens is you can keep getting the chemo, you'll keep going into remission, you keep relapsing each time faster, and then eventually the chemo will work. So they took a whole bunch of patients that had relapsed, they gave them the chemo again, they waited 3 months after giving them the chemo for the immune system to come back, because the chemotherapy also wipes out the immune system, that's an immune system cell. And they started vaccinating. And they did the same thing. They took the antibody that that person was making. They used each person as their own control. And of the 20 people that responded to the vaccine, not everybody did, right? Some of them are already tolerant. 20 people responded to the vaccine. Of those 20, 18 had not relapsed by twice the amount of time they had relapsed before, and by twice the average relapse time. One guy died of his tumor. One guy had minimal residual disease. I wrote to them and asked what happened to the guy who died. They said, we ran out of vaccine at 2 years. He died at 2.5. That paper was published in the Journal of the National Cancer Institute, and it sank like a stone. Is it because he's in Pamplona? I don't know. It's amazing. So, my advice to tumor people is, if you have a vaccine that seems to work, don't stop. Don't stop. Now, that's like the only thing you have to do. There are 4 things you need to do, I think. And I think, you know, the tumor people know most of this. This is not new to them. The first one, of course, is you have to raise an immune response. Not all vaccines work. You have to have a vaccine that actually gets you an immune response. 2, you have to make sure it's the right kind. And this is not new to them either. Right? IgE is an antibody that is used to get rid of worms. It's not going to kill a tumor. What you need to get is called a Th1 killer response. 3, you need to keep boosting. This, for some reason, is really hard. 4, you need to get the response to the tumor. Now, what we know is that once the killer cells are activated, you know, they leave that lymph gland, they go out into the blood, they start looking for cells that they can recognize. They're in the blood. And they don't leave the blood except in areas where the cells, the endothelium, which are the cells that line the blood vessels, have become activated. Right? They're just passing in the blood. At an area where the endothelium is activated, they will stop, roll, they'll slow down, they'll roll, they're kind of getting sticky. And eventually they stop, and they spread out, and they go through the blood vessel wall into the tissue behind it. You can vaccinate till you're blue in the face, and if you don't do something to the tumor to get the immune cells there, they won't do anything. With lymphoma, it's easy because the lymphoma is actually where the immune cells are. It's an immune cell. With a melanoma and a skin tumor, you need to do something to the melanoma. By the way, this is not new. There was a guy named Bill Coley in 1898 who was injecting bacterial toxins into tumors. And he cured about 30% of his patients that had sarcomas. Now, he was doing everything right. He was injecting the toxin to the tumors, he was doing damage in the tumor, so that when he got an immune response, the cells would come back to the tumor, and he just kept hitting them. He also killed a bunch of them in the beginning, because he was using living bacteria, in nasty one called erysipelas. But after a while, he started using dead bacteria and bacterial toxins. When he died, people kind of stopped doing it. His daughter started the Cancer Research Institute, and is still, pushing this Coley's toxin, it's called. Okay, so those are things. Now, I
What about
actually
getting rid of T-Rex?
T-Rex. Oh my goodness.
Negative rating.
Want to stick around? We'll talk about T-Rex. I'm not sure that everybody here. I actually don't think they're doing what most people think they're doing. Surprise, surprise. I think that what they're doing is switching the class of the response. Most people think they're actually dampening the response. And, but we can have that conversation later because a lot of people here. One of the things that Rosenberg is doing now is he's irradiating the shit out of people before he gives them pills. He's doing genetically engineered tills targeted to tumors. By doing that, he's got one paper saying he's now up to 40% response. Tumors are getting smaller. We don't know yet about cure. But it's not something you're going to do to, after that. It's very, very expensive. It's really hard on the patient. This thing is much better, I think.
Why can't they just cut out a tumor? Most tumors, for...
They can. And when they do, and they get it right, and there's no metastases, they can cure people. So surgeons have gotten better at that. That's one of the things that has gotten better since '71. Mostly because we're getting better at diagnosing tumors. So the surgeons can get to them, before they metastasize. But there's a really interesting model in mice where you inject this melanoma called B16 into the foot pad of a mouse, and it grows. The pad gets kind of big, and eventually you try to cure the mouse by cutting off the foot. And they do, they cut off the foot. Well, that's what they do with kids that have osteosarcoma. They cut off their legs.
What's that?
It's a bone tumor.
No.
And 25% of them are cures, and the other 75% die anyway because they've already metastasized to other parts of the body. But what happens in the mouse? They cut off the foot to save the mouse. The tumor goes to the lung. If they don't cut off the foot, the tumor doesn't grow. It's a very odd phenomenon. So I have this dog who's 14. She's had dogs get digital melanomas, they kill them. Oral and digital melanomas kill dogs. She's had a digital melanoma for the last 4 years. I've left her. What do you think influences what we do? She chews at it. Yeah, it's not getting too big. She's still alive. She doesn't have metastases in her bones. I get her x-rayed every year or so. What do you think influences what we do? I train sheepdogs. I have over 100 sheep. I train sheepdogs. I'm out in the sun a lot. Farmer's tan, I call it farmer's tan. And I get little basophiliomas on my nose. So I'm also nickel sensitive, as 10% of Western women are nickel sensitive. Oh, by the way, just as an aside, are any of you nickel sensitive? You can't wear nickel watches, some earrings, thank you. Yeah?
Earrings, really? Forget about it.
You know why?
No.
When you got your ears pierced, you use 14 karat gold posts.
Yeah?
14 karat gold has nickel in it. And what happens is you pierce your ears, they're damaged, sending lots of alarm signals, and that whole time that you're turning the posts, you're driving nickel in. And nickel binds to the peptides in MHC, and you've now become nickel sensitive. I got my ears pierced when I was 44 years old at a party with a lot of brandy and an 18-gauge needle. And before that, I could wear Levi's. And now I get this little rivet-shaped, response, like poison ivy response. So if you know anybody who's going to have their daughters or sons or whatever ears pierced, don't use 14 karat gold posts. Once they're set, you're fine. You're absolutely fine. Use platinum, silver, stainless steel, whatever, but not 14 karat gold. But anyway, all right, so I'm nickel sensitive. I became nickel sensitive at age 44. And so what I do when I get one of these little tumors is I go out and buy a cheap pair of earrings. I break off the part that goes through the ear. I take the flat part and stick it to my nose. I get this great nickel response. It's a very strong immune response. And the tumor goes away. And I don't have to go see a physician. All right.
What do you do if you're not nickel sensitive?
That's fetuses, transplants, and tumors. They're all still here. It's amazing.
Is the reason why monoclonal antibody therapies against tumors, the reason they work is because they're eliciting damage that...
Well, it depends on the antibody. So
HER2 or something.
HER2 is an interesting one. So the molecule that the antibody binds to gets dimerized by the antibody and signals. So it's not, you don't need other immune effectors. It's actually signaling the cell to die. And I don't know the exact, Johnson genes and stats and whatever that are involved.
But it's not damage that's causing an immune response to this targeted tumor.
No, not that one. No. Antibodies on the whole don't work on tumors. They're rare ones like HER2, new. And the reason that we know of has in the membranes of its cells molecules that counteract a whole series of molecules called complement. So antibody kills cells two ways. One, by this targeted assembly of a whole bunch of molecules called complement that eventually complement component 9 makes a little ring in the cell membrane and just suffers out. Amoeba histolytica does the same kind of little ring. And my complement will not work on my cells because my cells have several molecules that inhibit complement. Mouse complement will work on human, and human complement will work on mouse. Mouse doesn't work on mouse. So it's hard for an antibody to kill your cell that way. Now there is another cell called the natural killer cell, which is a killer cell. And itself doesn't have any specific receptors, but it can coat itself with antibody. And when it coats itself with antibody, and now that antibody is against the cell, the natural killer can kill that cell. It's another one of the arsenal that the immune system has. Ancient, that's an ancient cell. Sharks have them. Had enough? No? Want to do autoimmunity?
I just have lots of questions.
So I have a quick question about transplant, and that's actually to try to sneak in a question about parasites.
Okay.
So there's this sort of recent literature about virotype in transplants.
About who?
Virotype. So viruses living in our cells. What's that?
Virotype?
Virotype. So we all get various viruses, EBV, and so on and so forth when we're 3 or 4 years old or whatever. And then there are people that think if you transplant cells with the same virotype as the host into the host, that you're much less likely to have an immune response. And perhaps that has something to do with EBV and transplants. But there's not necessarily damage, and so what I'm wondering is, along with...
You can't do a transplant without doing it.
What's that?
You can't do a transplant without doing it.
Right, but in terms of healing, so you have, for example, if you have the baby hearts and the kidneys, right? But what if they weren't baby hearts? Perhaps if they were adult hearts and they fit somehow, which is possible. But the fact that they have viruses could mess things up. I mean, that's what these people think. And so what I'm wondering is, could there also be viral-specific responses? And the reason I ask that question is because I'm actually very interested in eukaryotic pathogens. And so I'm wondering if there are specifically, for example, fungal immune responses that look specifically for fungi, as well as damage.
Well, there are certainly immune responses to fungi. I'm not sure that they would respond to a fungus that doesn't do damage. And I know more of that in Drosophila than in mammals, right? So Drosophila has a particular, fruit flies, sorry, little fruit flies, have a particular set of genes that they use to fight fungus. But it's beginning to look like they're not actually recognizing the fungus as an initiator of the immune response. But when fungi get into Drosophila, to get through the outer coat, they use proteases and chitinases. And so proteases, for example, there's a molecule called spexel, which is broken off from a bigger molecule by protease. And that's a self-molecule that binds to the immune cells and turns on the immune cells. I think that, I call that an alarm signal. So if you didn't have proteases on fungi, I wonder if you would get immune responses. So I think that that's also damage and danger. Toxins, you know, people make antibodies to arsenic. You know how we've heard in I don't know, old English literature, old medieval literature, whatever, the kings who used to eat these poisons to get immune to them? Well, they were actually getting immune to them. They make antibodies. You make antibodies to arsenic and become immune to arsenic. Probably not all use toxins. So you can respond to those.
I have a question about adrenaline, like
Yeah.
that. So for me, then all of my studies with immune system and disease and wellness, that signal, that message, that thing that's in our body, adrenaline, that like if you can control that, there's a lot of just natural ways that you can help your immune system fight or not get illness. So I'm wondering about.
So adrenaline,
yes,
cortisone,
yes,
you know, stress molecules.
stress, like
Cortisone is an immune suppressant.
Yeah.
And when I was a graduate student, that used to really bother me. Because people said, you know, stress suppresses immunity. And I kept thinking, I always use the same example. You know, the gazelle that gets away from a lion is a pretty stressed animal. But it got away. But it might have gotten away, right? So it's now got 4 claw marks that it has to deal with immunologically. Bacterial infection, blah, blah, blah. So it doesn't seem evolutionarily smart for stress to be an immunosuppressant. But I just sat with, so I tell my students, you know, that the best mental condition for a scientist is confusion. Right? Because once you're confused, you're searching for things, right? When you think you know something, you stop, right? So I sat with confusion with that one for years. And then, because I like to go to odd immunology meetings, I went to a psychoneuroimmunology meeting in Florida. And there was a woman there from Denver, I can't remember her name, who asked exactly the same question about the gazelle and the lion. I couldn't believe it, right? Exactly, gazelle and lion. And she did these experiments with rats, not gazelles and lions, but with rats, where she acutely stressed them in various ways, right? So swimming stress, cat stress, that's the coolest one, where she would just line up her rat cages and put a little bit of cat food on each one and let the cat walk across eating the cat food. And it turns out that even the rats that have been in our research colonies since World War II, for generations they haven't seen a cat, are innately afraid of cats. So cat stress. And then after she stressed them, this is acute stress, after she stressed them, she injected them intravenously with a known number of E. coli bacteria and bled them 6 hours later and plated out the bacteria to see how well the immune system had gotten rid of them. And she found that acute stress actually caused the immune system to get rid of them better. It's long-term stress. Yeah. So then she started asking, all right, so what stress molecule is doing this? Is it adrenaline? You know, acetylcholine? I mean, what neurotransmitter that you might, you know, enhance by the stress is causing the immune system to work so well? It wasn't any of them. She adrenalectomized them, so it wasn't adrenaline. It wasn't any of those. And she found it was heat shock protein 70. So heat shock protein 70 has 2 forms, a constitutive form and an inducible form. It was inducible HSP 70 that was upregulated by the stress in the blood, that was now turning on the antigen-presenting cells that were engulfing the bacteria. Is that not cool? So the gazelle and the lion, the gazelle's doing well. It's that poor gazelle who's getting hunted by, you know, some guy on foot who's like walking after it every day after it, and long-term stress.
So would that affect somebody that has an internal thing that's constantly getting re-injured in a sense, aggravated or whatever?
So if you had something that was getting aggravated a lot, like consider like if I
I
started
have a
beating you
condition.
over the head with a baseball bat,
Well,
right?
I
Not
have
only would you
a-
be stupid, but you probably have an autoimmune disease in the head. So.
Well, not a bacterial, but I have hip dysplasia, so it's something that's constantly being aggravated. I can't do anything. There's
You
no.
gotta get arthritis.
Yeah, well, I know that eventually, but is there, with the body, what the body's doing with it, is there a way to get it to stop?
The arthritis?
No, stop.
Well,
Stop
the
that. I
arthritis
mean, the
is
pain
immunological.
and all of this. You know, suppress, I guess more suppress that way.
I don't know enough about pain.
Yeah.
Yeah, I don't. You know, with dogs they do surgery.
Yeah, I've already had surgery. I've had surgery.
Yeah, I don't know.
Yeah.
There are ways to slow down the arthritis, because the arthritis is going to be, you know, immunological.
Mm-hmm.
The problem with it is, I mean, there are people, for example, who have rheumatoid arthritis, and they give them a soluble receptor for one of the molecules that's involved in that called TNF, tumor necrosis factor. And these people get off the table saying, "Thank you, doctor." Right? because it works so quickly to relieve their pain and so on. The problem is that they're now prone to viral infections. You gotta be really careful when you use general immunosuppressants. Sorry.
No, I was just curious.
Actually, I had a really good friend who has hip dysplasia and had 4 kids, and all of them had autism. You guys done?
No, not at all. I think maybe if people want to stick around and talk to you, that'd be great. We're here as long as you want.
Well, I could do autoimmune.
But if you want to get up, there's
about,
snacks.
It takes about 15 minutes. So who would like to go through autoimmune? Okay. There's some of you who are still sitting there who didn't want to do that. Is there something else you'd like to do instead?
Now's the chance.
Yep, now's the chance to vote with your feet. So does anybody here have an autoimmune disease?
Crohn's considered autoimmune.
Well, you don't have one yet.
My wife has type 1 diabetes.
That's autoimmune.
I have one.
Well, sorry?
I have Crohn's, and I'm on
Crohn's.
a Humira. It's called. Is the drug that I'm taking?
I don't know the brand names. Do you know what
It's It's a tumor, or it's a TNF, I think.
Anti-TNF?
Yes.
Probably anti-TNF.
Yeah.
Yeah.
It's what they use for rheumatoid arthritis.
Yeah.
I'm surprised.
It's probably a soluble TNF receptor.
Yeah, it's
Does it work?
yes, it's worked miracles actually.
Great.
It's
Yeah, you're in my 5th category of autoimmune. All right, so let's do autoimmune. So before we do autoimmune disease, I want to remind you of 2 conditions that are not considered autoimmune disease, because we know what the antigen is and the disease stops when the antigen goes away. And the first one is rheumatic fever. So this is something that kids get. So most of us, if we get streptococcus, clear it without a problem. Every so often you get somebody that when they're making an immune response to strep, the antibodies to strep also cross-react on the heart. And while they're fighting the strep, they're destroying the heart, usually the valve. So physicians who take care of these patients tell me the best thing they can do is to put them on a ton of penicillin and get rid of the strep as quickly as possible. Because when you get rid of the strep, the autoimmune response also goes away. So what that says to me is that, remember I said the immune system doesn't throw fuel on the fire, you need the real damage. So with this autoimmune phenomenon, when the bacterium goes away, the autoimmune response stops. So you need the real damage, the real danger, the real pathogen. The second one is celiac, which is different from Crohn's. And what's happening there is, it's really not autoimmune. It's a response to a protein in wheat. It's a peptide that people call a toxic peptide, is gliadin, which is a component of the gluten of wheat. And it's not autoimmune, it's just that some people respond to this toxic peptide by making an immune response type, which is toxic to the gut. Right? They make TNF, IL-15, interferon gamma, oxygen radicals, activated macrophages, and what happens is the villi of the gut flatten. They can't deal with this. And so because of diarrhea, they have inflammation, they starve, and all that has to happen is they stop eating bread.
Were you born by C-section? Were you born by C-section? Do you know about the theory about people that are born by C-section?
Have a higher propensity to have?
Yeah.
I've never heard that one, no.
I don't.
Interesting.
Yeah.
Isn't that's related to microbiota, right?
Yeah.
That's interesting because most people I know who've got celiac tend to get it fairly late, not early. It's also underdiagnosed, well, it used to be underdiagnosed hard. I have a friend who had it for 12 years before they finally figured it out. They sent her to shrinks and the medical system does not treat women. And then you get, but of course, you guys don't have the politicians in your life. At least not to the same extent. Let's not go there. So, remember those two diseases, right? One is not autoimmune at all, but what it says is that the wrong kind of immune response can destroy a tissue, whether that response is directed at the tissue or at some pathogen in that tissue. And also that you need the real pathogen to keep the immune response going. That's what rheumatic fever does. Okay, so let's do autoimmunity. Autoimmune disease. So there are people who are trying to clone genes for autoimmune disease. To them, there's this umbrella called autoimmune disease. And they're going to clone, I guarantee you, they're going to clone every gene involved in an immune response and then some. I think there are 5 categories of autoimmune disease. And in 4 of them, there's nothing wrong with the immune response. So I didn't make all these up. I made one of them up. I've modified most of them. But these are categories other people have also suggested. So the first one is boring. It's really boring. You have an infection and you don't know it. So if I give you a mouse dying of a virus called lymphocytic choriomeningitis virus, and you didn't know there was a virus there, you'd think it was dying of an autoimmune anti-brain disease. Because what happens is LCMV, the target, the main target for LCMV, certain subtypes at least, is the choriomeninges, the blood-brain barrier. So it infects those cells, and then the killers go and kill the infected cells. And so they open up the blood-brain barrier. It's not autoimmune. But if you didn't know that there was a virus there, you'd think it was autoimmune. So that's LCM, that's in mice. And my favorite disease here is Lyme in humans and dogs and whatever. So Lyme, when it first happened in the town of Lyme, was diagnosed as juvenile rheumatoid arthritis. That's what it looked like. Now, juvenile rheumatoid arthritis is a pretty rare disease. What happened is that there was this epidemic of arthritis going on in these kids in Lyme. And the CDC, well, he had been in the CDC. This young guy named Steer, S-T-E-E-R-E, who had been at the CDC, went up there to have a look. And he stole a nurse's observation that a lot of these kids lived near the woods. Actually, how many physicians know the addresses of their patients? But anyway, he became famous. He became famous and suggested that it was really an infection. And people looked for the infectious agent for a long time. And finally, a guy at the NIH in the Rocky Mountain Lab figured out that it was a spirochete. It's kind of bacterium. It's called Borrelia. It's not juvenile rheumatoid arthritis. So you have an infection, you don't know you have an infection, your immune system is doing its job, it's clearing the infection, but in the process, it's causing damage. You know, the immune system is an amazingly powerful system. It can kill. It does kill people. People who have major trauma, you know, stepped on by an elephant, hit by a car, 50% of them who come into emergency rooms with major trauma die, not of their trauma, but of something called systemic immune response syndrome. They go into multi-organ failure. Their immune system is on. Everything is on. The immune system is responding as though they had a massive blood infection of bacteria. But the physicians can't find bacteria. And there was a guy at Harvard, a trauma surgeon at Harvard, who's also a rock musician, who still plays in a band, who discovered that it's the release of mitochondria from the damaged tissue. Remember, mitochondria are these ancient bacteria. And the immune system is reading this as a massive bacterial infection, and it kills you. So it's an immensely powerful system. So, first category, you don't know there's an infection. I have psoriasis on one elbow, but only on one elbow. And I keep it down by using those antifungal foot creams that people use for athlete's foot. I have a very good friend who's a dermatologist He says, it can't be fungus. I say, why? He said, we would see the fungus in biopsies, right? Because fungus make these little rhizomes, right? So, I think psoriasis may be here as well. One of the things that's interesting is that some people get fungus on their toenails, and foot doctors know that what happens is usually you get some kind of injury, and that's where the fungus goes in. Okay? And it's interesting, if you look at psoriasis, it very often happens in places that get injured. Elbows, sunburned areas, and so on. Okay. The second category is similar, but different in a subtle but important way. And that is something that people call molecular mimicry. You have an infection, and the response to the infection gives you autoimmune response. So, rheumatic fever is here. Right? You make an antibody to streptococcus, and the antibody also sees a heart valve protein. Now, people say, well, why do you have B cells that can make an antibody against your heart? They should be gone. You should be tolerant. People in autoimmunity have been asking the question, how do you break tolerance for the last 60 years? And I say you can't break what doesn't exist. I think we are never tolerant. As long as the thymus is putting out new T cells and the bone marrow is putting out new B cells, we are never tolerant. We're always in the process of becoming tolerant. Now, some tissues are better at doing that than others. So for example, if you have a T cell that leaves the thymus and comes out, it has the ability to see your own liver. The liver is huge. And the blood vessels that line the liver are fenestrated. They have little windows so the cells can leave easily. And the T cells circulate the body about once every 24 hours. So within a few days, the T cell that has left the thymus is specific for liver will have gone through the liver and gotten tolerized. But a T cell specific for brain is in a different situation because the brain is barricaded. Now, people used to think there was no traffic through the brain, but that's not true. There is, but very little. So a T cell that leaves the thymus specific for brain is going to probably circulate for a long time before it manages to get through the brain and get tolerized. A T cell specific for islets of Langerhans are tiny, and they're barricaded. So T cells against islets are going to circulate for a long time before they get tolerized. No longer surprises me that a lot of autoimmune diseases are against barricaded or small endocrine organs.
So if that's true, would you get autoimmunity if you had a chronic wound that was multiple?
Absolutely. And in fact, there's something called chronic autoimmune hepatitis, which actually turns out to be hepatitis C virus. But, you know, that's a chronic virus. It's constantly doing little damage. And you can also get an autoimmune reaction out of it.
I feel like do diabetics get it?
The problem with a diabetic is you're not diabetic until you've wiped out 90% of your islets. So, and they keep getting wiped out. So eventually you've wiped out all of them. They're gone. You can't make them again. And people are working on how to give duct cells the signals to make them again. You can't make them again. So it doesn't matter that you're still autoimmune. You could never get tolerized. Your islets are gone.
No, what I meant was their chronic wounds that diabetics, they would get response, autoimmune response to that.
Nobody at the moment knows what causes the immune response that gives type 1 diabetes. In fact, I'm teaching a course on a class on autoimmunity next Tuesday at the NIH, and I gave the students a whole bunch of papers to read about what causes diabetes. Because nobody knows. In fact, for most of these autoimmune diseases, people don't know. Rheumatic fevers are really unusual. Okay, what's different here? They're both infections. But here, there's no autoimmune component. The killers are killing cells that are infected, which means the infection has to be in the target organ. Here, there actually is a cross-reaction. Right, a B cell came out of the bone marrow, it hadn't gotten tolerized yet, it got turned on by strep, and it'll keep getting turned on as long as the strep is there. And by the way, if you put these people on penicillin for 20 years, you can then stop because they become tolerant. But it takes time because the bone marrow is putting out new B cells every day. So here, the infection need not be in the target organ. You could have a gut infection that cross-reacts on the brain. And here there is an autoimmune component, and here there isn't. But in both cases, the immune system's doing its job. It's fighting an infection. All right, category. You can't have under a self-not-self model. You can only have it under a danger model, and that is bad death. So the danger model says that good death doesn't get you an immune response, bad death does. I think lupus is here. Programmed cell death is a very complicated process. It was first discovered in a worm called Cinerabdatus elegans, this beautiful worm. And elegans because it is, you know, elegant. In the worm, there are 19 genes that govern death. 11 of those are about cleaning up the mess, scavenging, and so on.